Hi everyone, welcome to the RNA algorithms course. My name is Eugene Baulin. I'm the PI of the Laboratory of RNA Algorithms at the IMol Institute, or in full, the International Institute of Molecular Mechanisms and Machines of Polish Academy of Sciences. We're located in Warsaw. And this course is read at the Faculty of Mathematics, Informatics and Mechanics of Warsaw, of the University of Warsaw. And this recording is from the semester 26-27, the winter semester. So this is the first lecture, and first of all, this is the more correct title of this course. So by RNA algorithms, I mean algorithms and computational problems of RNA structural studies. So we will cover a range of computational approaches for studying RNA sequences, RNA secondary structure, RNA 3D structure, their predictions, annotations, comparisons, or some other computational problems that we have in this field. This is the first lecture, the introductory one. So first I will briefly recap the history of RNA, our knowledge about RNA structure, and then I will briefly explain what this course is about, so what to expect, what will be the format, and how you are expected to pass this course. So let's start. So we are humans, right? And in this weird plot you can see our human body composition. So we know that water is roughly 60 or 65% of our body, and other than that we have organic matter, and among this organic matter we have proteins, which are the most most excessive amount of this organic matter, and nucleic acids compose very small amount of our body, but they're very important. And largely this course will focus on RNA, but we will mention in some regards proteins and DNA as well. So here I encircled proteins and nucleic acids as linear biological biopolymers, which consist of monomers, like amino acids in case of proteins, or nucleotides in case of nucleic acids. So these are the objects that we will focus throughout this course, but mainly, of course, we will focus on RNA, which is a nucleic acid. Now, you may heard of the central dogma of RNA biology. So currently, this is more or less the modern scheme of the dogma. So we do have DNA, so this is the dogma about the information flow in molecular biology. So we do have DNA, which is the storage of our genetic information. It's usually forms a double helical structure, as shown here. We have RNA, which is in the case of messenger RNA. This is just a proxy between DNA and protein. So the RNA is a single-stranded molecule, but it does not look like this. So this is what a helical fragment of a single-stranded fragment of a double helix looks like. So like a half of this double helix. So RNA, of course, does not form this absurd structure, but it's single-stranded, and it's kind of like DNA. So that's why this is how people depict RNAs often. So both DNA and RNA are composed of 4 monomers. Nucleotides, so deoxyribonucleotides in case of DNA, or ribonucleotides in case of RNA. So for DNA we have ACGT for possible nucleotides. For RNA we have ACGU, so instead of thymine we have uracil in case of RNA. And the process of obtaining RNA from DNA is named transcription. The process of obtaining proteins from RNA is called translation. So if some of you may know that ribosomes are responsible for the translation, and this is how we depict proteins. So they the globular proteins. Proteins consist of 20 different monomers or amino acids. So it's completely different alphabet from DNA and RNA, and completely different chemistry. And so we also have the processes of duplication of DNA and RNA, which are called replication. In some weird scenarios, we have retrotranscription, which is building DNA using RNA template. And the central dogma actually states that once the information is encoded in proteins, it cannot go back to DNA. So we don't have molecular machines that perform the decoding of protein information back into DNA. So that's the central dogma. Of course, like in biology, we always have exceptions. So recently there was a paper about proteins that synthesize some repeated DNA sequence, like some trivial sequence composition, without any templates. And formally, this is building DNA from protein. It's not yet like a flow of information, but maybe no one is sure that at some moment we will have this arrow identified and named. But for now, it does not exist. Okay. So these are the three main objects that we consider. And of course, we will focus on RNA, which is not only a proxy between DNA and protein, but we will focus on structured functional RNAs, which we have a lot of currently. So how it all started. Of course, now we know, like, we have DNA, we have RNA, this is how the structure are organized, the monomers, etc. But originally, it all started back in roughly 1869. So these are the two screenshots from a paper written in German by Friedrich Mischer. So Friedrich Mischer isolated substance from pus cells nuclei, and so he actually isolated the content of nuclei of pus cells, and he characterized it. He actually spotted that this substance behaves differently from proteins. So proteins people were familiar with back in the days. So he noticed that this substance contains nitrogen and sulfur. Contains nitrogen and sulfur, but is very rich in phosphorus, and phosphorus is absent in proteins. Of course, we now know that sulfur is absent from nucleic acids, so it's the isolation was not perfect, presumably. So, and he coined the term nuclein to describe for this substance that he isolated. 20 years later, his PhD student renamed this substance into nucleic acid, because he apparently spotted acidic features of this substance. So we had the term nucleic acid from roughly 1889, and we didn't know the difference of DNA or RNA. We just knew that we have nucleic acid, which is different from proteins, and it is located in nucleus, cell nucleus. So then, at the moment of early 20th century, actually people distinguished two types of nucleic acids, DNA and RNA, but at that moment the differences were not apparent. So people isolated DNA from thymus and RNA from yeast, and for some time people assumed that DNA is actually animal nucleic acid and RNA is a plant nucleic acid or something similar. Then in 1919, Phoebus Levine hypothesized the content of the nucleic acids. So he mentioned that, stated that they are supposed to consist of 4 monomers: cytosine, thymine, which are pyrimidines, the 6 member rings, and purines, guanine, and adenine. And the structure of the single nucleotide is looks like this. So we have a base, we have deoxyribose sugar in case of DNA, or ribose sugar in case of RNA, and a phosphate. And now we know that this is true, so this is a true composition of nucleic acids. A bit later, in 1933, people got the idea that our DNA and RNA are not from different life kingdoms, but actually DNA is mostly located in nucleus and RNA is mostly located in cytoplasm, and so this is correct as we now know. So mostly RNA is located inside the cytoplasm, indeed. But until middle 20th century, people were mostly interested in proteins because those were the live molecules, the most interesting ones, and DNA and RNA were just boring. But these two experiments from 1944 and 1952 turned the tables. In 1944, Avery, MacLeod, and McCarthy showed. So they did what they did. They used mice. They injected virulent, so pathogenic bacteria into mice, and mice died. Then they injected non-virulent strain of bacteria in different mice. Mice didn't die. So we have like a pathogenic, deadly bacteria and innocent bacteria. Then they heat-killed they fried the pathogenic bacteria. So what we have, by heat-killed, we destroy all the proteins in bacteria, and they injected the mouse. Mouse didn't die, but then they also used this heat-killed pathogenic bacteria, which is like everything but proteins. Proteins are destroyed, and the non-virulent strain of the same bacteria. And when injected together, mice died again. So which means that something in these virulent strains, which are not proteins, were responsible for the virulence of this bacteria, which means it's not proteins, right? But this was not decisive. So people were a bit skeptical about this experiment. But the second experiment of Hershey Chase was much more straightforward in communicating the importance of DNA. So what they used bacteria viruses, which are called bacteriophages, in two types. So in first, they labeled sulfur atoms of proteins of these bacteriophages, and they monitored how many of this labeled sulfur will come in to the infected cells. There was none, so no sulfur comes to the cell from the bacteriophage. And then they alternatively labeled phosphorus of DNA in bacteriophages, and they spotted that DNA is being injected in the infected cells. And this was a clear communication that actually DNA is responsible for the replication of viruses, and it means that DNA is actually a storage of genetic information. And since then, everyone was interested in DNA. DNA became the molecule of life, and people were trying to analyze it and pretty much resolve how its structure is organized. And in 1953 was the very important year, because it resolved the competition of identifying the structure of DNA. So the primary contestants were the Watson and Crick and Linus Pauling. So Linus Pauling at that time already correctly identified the alpha helix structure of proteins, and so he was very famous at the moment, and everyone knew that he wanted also to identify the structure of DNA. And Watson and Crick were not famous at the time; we were very young scientists, but they had two key advantages. First of all, they had great crystallograms of Rosalind Franklin from Wilkins' lab. This is the crystallogram of a B-form DNA helix. This is how it looks like. And so here you can appreciate how this crystallogram translates the parameters of double helix of DNA translates to this crystallogram. So it's not at all trivial to recognize how the helix looks like from this crystallogram. And unfortunately, Rosalind Franklin herself was interested in A-form DNA, because it was a common assumption that A-form DNA is a biologically relevant conformation, and B-form is just like an artifact of crystallization conditions, which now we know is not true. So we know that B-form helix is a biologically relevant structure. And Watson and Crick also were friends with Chargaff, who introduced his famous Chargaff's rule that the content of adenine in DNA equals to the content of thymine, and content of guanine equals to the content of cytosine. So knowing this rule, so having this rule in mind, having these perfect crystallograms, they played with the real models of DNA, and they correctly resolved the structure of DNA, and actually got the Nobel Prize a bit later for this. So but it was not like the end. It was just the beginning, and RNA was not in the game yet. So now this paper on the left is an interesting one. So it's by Belozerski and Spirin from 1955, published in Nature, and it stated that in different organisms we see that the nucleotide content, like the GC content, so how many more we have guanine and cytosine over adenosine and thymine, differed from organism to organism. So in some organisms we have like high GC content, in some other organisms we have low GC content, so it differs from organism to organism. Whereas the RNA content they measured, and it did not correlate with the GC content of DNA. So for RNA, the GC content was pretty much static across organisms. So it was like surprising. So how RNA is supposed to be? So if we assume that RNA is a proxy between DNA and protein, then RNA encodes the same message, the same genetic information as in DNA. So the content should be the same. But it was not the case. Now we know that actually in mass, ribosomal RNA dominate the RNA content in cells, and ribosomal RNAs are the most conserved RNAs, and so in all organisms we have more or less the same ribosomes, and their nucleotide content is static, and so they compose up to 90% of all RNA in cell. But at that moment they didn't know this. So a bit later, so yeah, as I said originally, I didn't say that yet. So originally the central dogma was coined by Francis Crick in 1957 or 58. It was a statement that information flow doesn't go back from proteins to DNA. And actually the path, how we know it now, from DNA to RNA and RNA to protein, was coined by James Watson, I think, by Francis Crick, sorry, in 1960. And then messenger RNA was identified in 1961 by two different groups. One of them was the James Watson group. Yeah, so RNA came to play only around 1960. But before it was not that obvious what RNA has to do with DNA and proteins. Now then, this is paper of Fresco Albertotti from 1960, and this is, as I understand, the first drawing of RNA secondary structure. As you don't know yet, but so in DNA, this double helical structure is composed of base pairs, and the canonical Watson-Crick base pairs are in the canonical Watson-Crick base pairs, G pairs with C and A pairs with T. The same with RNA. So RNA is composed of these like short helical fragments, but in entirety it's a single strand. But some fragments form these helical regions, and we also have G pairing with C and A pairing with U instead of T. And this is the first drawing of RNA secondary structure, which is a set of canonical base pairs. Now we know that it's not, so RNA secondary structure not exactly look like this. We don't have this much of bulged out bases, so the majority, the wide majority of base pairs actually are consecutive with each other, so the amount of this single bulges is not that high as it was thought here. Then another interesting paper from Spirin from 1960, where he, as I understand, for the first time coined the terms primary structure, which is an RNA sequence, secondary structure, and tertiary structure, like distinguish three levels of RNA structural organization. So where primary structure is a sequence of nucleotide alphabet, Secondary structure is a set of base pairs, as shown here, and tertiary structure are pretty much the spatial structure. And another interesting statement, interesting paragraphs from this paper is that at that moment people asked the question whether RNA in some complexes that they found RNA in, so in RNA viruses or in ribosomes, which consist of RNA and proteins, whether the RNA content comes as a single polymer chain, a single long linear polymer chain, or it's a set of different chains. And funny thing that Spearing and his lab worked with viral RNAs from viruses, and they worked with those viruses in which genomes are indeed a single RNA strand, a single continuous RNA chain. And Doty and his lab worked with ribosomal RNAs, and they, in contrary, identified that a ribosomal RNA is not a single polymer chain, but actually a number of different chains. And this was presumably in conflict. So the conclusions of the two labs were in conflict with each other. So one lab showed that it's a single chain, and the other showed that it's a number of chains. But now we know that actually yes. So in ribosomal RNAs we have three, or 1, 2, 3, 4. No, from organism to organism, three or four RNA chains in ribosomal RNAs, and one chain in actually single chain RNA viruses. So we do also now know the RNA viruses which have their genomes in a number of separate strands, but the Spearing lab worked with single-stranded genome viruses with single strands of RNA. So these were the examples of knowledge that was not always there, and now we know it. But at that moment, it was not. It was pretty many more things were unknown back in the days. So then, the first RNA sequence of a tRNA, of a transfer RNA. This is an RNA that brings amino acids to ribosomes during translation for building new proteins, if you don't know yet. So the first RNA sequence of a tRNA was identified in 1965, and the paper featured in figure 2 of this paper, they featured three alternative predicted secondary structures for this RNA. So they identified the sequence, but they didn't know what the secondary structure of this, and the 3D structure was also unknown back then. So they hypothesized three structures. Now we know that actually this one is the correct structure, which is a famous form of a clover leaf structure, because it looks like a clover leaf. And these are the incorrect ones. So then the 3D structure of the same RNA, the same tRNA, was resolved using X-ray crystallography in 1975, 10 years later. So this is the density map that was resolved using crystallography. This is the backbone trace, so the trace of the backbone backbone skeleton of the molecule. So these are all the phosphates and riboses connected to each other of the consecutive nucleotides without showing the bases. This is the secondary structure derived from this density map. So we know, so people saw that this is indeed the cloverleaf secondary structure. People named this 3D structure form as a L-shaped. So because if you rotate it, it will be L. So it's organized like this. So we have actually these four stems. So stem is a stack of consecutive base pairs. And we have four stems, and they are organized in two pairs of coaxially stacked stems. So these two stems coaxially stacked with each other, these two stems coaxially stacked with each other. And in this, between these two loops, we have some long-range interactions that let the molecule assume the L shape. So now we know all this. So now we actually have thousands of known RNA 3D structures, and at 1975 this was the first one. And I imagine how hard was it to work with this with all this data of crystallography without modern computers. So it was I assume it was a nightmare. Okay, then Around 1975, people knew about messenger RNAs, people knew about ribosomal RNAs, viral RNAs, and transfer RNAs, but still all these RNAs were involved in the translation of proteins. So RNA was still like a intermediate for protein translations and nothing more, until in around 82, 83, two back-to-back papers came, one from Thomas Cech lab from Colorado and one from Sidney Altman group from Yale, which showed that actually without any proteins, RNA is capable of catalyzing reactions. And Thomas Cech worked with group 1 intron of tetrahymena, and the function of this intron is to self-cleave itself from flanking sequences. So just like the intermediate sequence within some like a genomic context is forms some structure and cleaves itself from it. And ribonuclease P is involved into processing of tRNA genes. So it cleaves not itself but some other RNAs. So they showed that these two RNAs are capable of performing catalytic reactions, which means that they are functional molecules and don't depend on proteins for this function. And so since then we know of the concept of ribozymes. So ribozyme is like a merge of two words, like enzyme, which is a catalytic protein enzyme, and ribo stands for like to show that it's RNA. So RNA enzyme is a ribozyme, which translates to catalytic RNA. And they got the Nobel Prize together in 1989. So it was the start of the era of functional non-coding RNAs, structured molecules which have nothing to do with protein translation, but perform some other important functions in cells. So now we know a wide spectrum of non-coding RNA types. So just a handful of types of like large groups shown here on this slide. So we have ribozymes, as we already saw. So ribozymes perform some catalytic reactions, and they need their structure to do this. Riboswitches also they usually regulate some processes via conformational changes. So one canonical example is a ligand binding riboswitch. So once we have no ligands, the riboswitch forms some structure. Actually here, so here it's the opposite. So once bound to a ligand, riboswitch has one structure. Once we have a deficit of this ligand, so we don't have a bound ligand, the structure differs, and it leads to the formation of different structures in the nearby regions, which either like block or in opposite unblock some translational transcription or some other mechanisms, or some binding to something. So the concept of riboswitch is that it switches its conformation based on binding to something, and the switch of conformation regulates some following processes. So some riboswitches even change the structure not by binding to a single ligand, but by sensing some like environment conditions, like the metal ion concentrations or temperatures. We have even temperature sensors of that are RNA. Small RNAs of different kinds, the most famous I think like microRNA, which also bind to some messenger RNAs or some different DNAs as well, to again regulate like which genes we will turn on or off, and what protein products will be produced. Long non-coding RNA is one of the most recently discovered types. The only common thing of long non-coding RNAs is that they are long and they are non-coding, and that's it. So we have a spectrum of different types. We don't know much about them, so like in general. So we do know something about some specific long non-coding RNAs are involved in some like cancer diseases or something. But in general, we don't know much. But we know the overall concept is that for these long non-coding RNAs, they do not adopt a single like rigid 3D structure, but rather they have a set of like sequence domains, sequence motifs, structure domains, and some of this structure are responsible for binding to something. Some of the sequence motifs are responsible to binding to like proteins or DNA, like in a complementary manner, or something else. And so like long encoding RNAs is just like a string of bits, and different bits are responsible for different like subfunctions of these huge molecules. So the even more recently discovered are circular RNAs, which are not a linear polymers but circular molecules. With like one of the discovered functions for circular RNAs serving as a microRNA sponges. So like this circular RNA compete for microRNA binding with messenger RNAs and like make these microRNAs bind to them instead of the target messenger RNAs. And now we do have more and more types being discovered each year. So this is really just a set of short list of examples, but not a comprehensive one. So now we know a lot of different RNAs, but for some of them we don't know much. So for some we know for a minority we know the structure and the function, and for the majority we know neither the structure nor the function. For some of them we know only the function, for some of them we know only the structure. So now about this course, how this course came to life. So originally I was studying RNA structures at the Institute of Mathematical Problems of Biology of Russian Academy of Sciences in Pushchino, by supervised by Mikhail Roitberg, who is sadly not with us anymore. And he suggested me to read bioinformatics course, like a hybrid bioinformatics algorithms course developed by Pavel Pevzner and Philip Kompu in 2014 from University of California, San Diego. So Mikhail Roitberg suggested me to read the hybrid form of this course at the Moscow Institute of Physics and Technology, and I read this course from 2015 to 2021. And so like I learned the structure, like the structure of the course I learned by heart, and I try to re-implement the same concepts into this course. Then I moved to Warsaw, Poland for my postdoc at Janusz Boniecki lab. So I was involved in primarily like tool development regarding RNA structure comparison, RNA structure modeling and prediction at IMCB in Warsaw. And since 2024, I'm a PI of Laboratory of RNA Algorithms at IMol, Polish Academy of Sciences. I'm continuing to work in prediction of RNA structures and in the different other computational problems of RNA structural studies. And since 2025, I read this course on RNA algorithms at the Mathematics, Informatics and Mechanics faculty of University of Warsaw. So how this course, what are the objects that we will be covering? So we will mostly cover 4 types of objects of RNAs, so which are like different levels of RNA structural organization. So one of them is RNA sequence, which is just like a string in 4-letter alphabet, AUCG. It's a linear one, it has a start and the end. And if we have a set of homologous RNA sequences, or like similar RNA sequences, we can align them, highlighting the corresponding regions, which can hint us some information about the conserved parts of the sequence. Either having sequence or sequence alignment, we can try to predict RNA secondary structure. So secondary structure is another object, which is basically a set of canonical base pairs. So an RNA sequence and a set of canonical base pairs. These are 3 ways how we usually depict secondary structure on a plane. So this is the RNA secondary structure graph. So these blue edges are base pairs, and these gray short ones are covalent bonds between consecutive nucleotides. And alternatively, if we stretch the sequence left to right and depict the base pairs as arcs, then we will have arc diagram, or as if we depict base pairs as pairs of matching brackets, parentheses, we have a bracket diagram. So this is famously named dot bracket notation. And of course, another object is RNA 3D structure, which is basically like a text format file of atom coordinates. So we have two major 3D structure formats, PDB and MMCIF. So PDB is a legacy format of PDB databank, and MMCIF is a new PDB like protein databank format, which became the main format from 2014. But until now, the majority of tools, of all tools especially, work with PDB format only, and pretty much all the new tools work with both formats, but still both of them are like slightly different versions of text form. So, and the arrows here show like the computational problems that come across this objects. So once we have a set of homologous sequences, we have a computational problem of aligning them correctly. Once we have sequence alignment, we, for example, have a problem of deriving the profile of sequence alignment, or like the centroid sequence, or something else. If we have a sequence, we can predict secondary structure. If from sequence alignment, we also can predict secondary structure, and it will be a bit more. We may be a bit more confident in these predictions, but it's like it differs from sequence alignment. We predict the conserved secondary structure. From RNA sequence, single sequence, we predict like this secondary structure of this sequence, which will be adopted by this sequence. Like in the opposite direction, having the structure may help us to align the sequences better, or having the structure, we may ask a question of like, what will be the sequence that will adopt this structure, for which this structure would be the most optimal one. So like this would be the RNA design problem, like how to design a sequence for the specific structure of interest. And of course, having alignment or RNA secondary structure, we may want to predict 3D structure, and on the opposite, having 3D structure, we may want to annotate what are the interactions there, including canonical base pairs, which would be the secondary structure. Or RNA 3D structure may show us also the hints of realigning correctly the homologous sequences, right? So sequential information on 3D level may also help us improve RNA sequence alignments. So these are the main objects, and these are the computational problems that we will cover. Mostly like comparison, prediction, and analysis, right, of RNA structural studies. So this is my favorite example of a computational problem. Just for you to get an understanding of the problems that we face in RNA structural studies. So this is the input of this problem is two arbitrary RNA structure fragments, which is mathematically speaking just two sets of residues. And the output of the problem is to find the correct, like the best superposition, or the correct one if we assume that the correct superposition of the two arbitrary fragments exists, right? So we want to identify whether these two fragments are similar or not, and for that we need automated methods of finding the best superposition. So in general, like this superposition is a partial matching between the residues of the two fragments, and having two sets of residues, the problem of finding the partial matching is a factorial complexity problem. And of course, factorial complexity is a bad one. So we will cover in our course how we develop algorithms to overcome such complexities, but still solve the problems in the efficient way. So what content we have in this course? So we have 15 topics, which I will read in 13 lectures at the university. So the first one is this one, introductory one. So the second one is in general about algorithms and computational problems and what defines a good computational problem. Then I will cover in the third lecture, I will cover like the terms and the features of RNA structures, the file formats, some elements of structures, representations, databases, like pretty much all the context of terms and locations and formats that we have, and you need to know of to work with RNAs. In lecture 4, we'll describe like what types of experimental data we have from modern experimental techniques, which actually define the computational problems. So if so we don't have, for example, now the problem of predicting RNA sequences of our genome, because we can just sequence them, and that's it. So the experimental techniques actually define what computational problems we will have to solve. So then we will switch to 3 levels of organization. So the algorithms working with RNA sequences are covered in lecture 5. Then we have 3 lectures, 6, 7, 8 on RNA secondary structure prediction and different settings. So in single sequence output, input, alignment input, with or without pseudonodes, which is a special type of RNA secondary structure element. So 3 lectures for this. Then 1 extra lecture on secondary structure prediction, covering some like exotic topics, including, for example, co-transcriptional folding. So what happens if the RNA secondary structure folding happens simultaneously with RNA sequence formation during transcription, how it changes things. And then we will devote 5 lectures to 3D structure studies. So first 2 would be about 3D structure annotation and 3D motif search, which is pretty much the same thing, but annotation is a broader topic, and 3D motif search would be focused on motifs, like on the recurrent 3D motifs of RNAs. Then lecture 12 is about global alignment of 3D structures. Then lectures 13 and 14 about RNA 3D structure prediction. So we'll cover different modes when we do have homologous templates and we have no good templates to base our predictions. And again, some in lecture 15, some exotic topics on RNA 3D structure prediction, covering some like artifacts, scoring functions, and some other techniques, not conventional ones. So that's all for the first introductory lecture. See you in the next one.